Assalamualaikum and salam sejahtera. My name is Armania Nurdin and I'm from Universiti Putra, Malaysia. Before we go further, I would like to say thank you so much to MAKNA for giving me this prestigious award. I'm really grateful for this wonderful opportunity. So this is the research title that our team have proposed for MAKNA Cancer Research Award 2019. Elucidating the roles of circulating exosome protein biomarkers in prostate cancer cell invasion and migration. In prostate cancer, one of the major unmet critical needs is the ability to distinguish between aggressive and non-aggressive prostate cancer. So if we can find a suitable or ideal biomarker that can distinguish between these aggressive and non-aggressive prostate cancer, so the doctor might be able to determine effectively which patient should receive immediate treatment or undergo active surveillance. So this will eventually prevent the over-treatments of the non-aggressive prostate cancer. So early detection of patients with clinically significant prostate cancer is really important for us to give immediate treatment to inhibit the progression of those prostate cancer. For example, which of these men predicted to have potentially aggressive and later prostate cancer? So in prostate cancer, 1 in 7 of these men will be predicted to progress to metastatic stage of the prostate cancer. For example, bone metastatic prostate cancer. And majority of these men will remain with localized and non-aggressive prostate cancer. So majority of these men could live with this slow-growing prostate cancer for the rest of their life without encounter any significant related complication. And however, these patients usually suffered due to the treatment-related complications such as urinary incontinence. So an ideal biomarker or serum marker should be able to identify which of these patients have high risk to develop metastatic stage of prostate cancer, so that an immediate treatment can be given to these patients and to inhibit the progressions of the prostate cancer. At the present moment, prostate-specific antigen or PSA is a protein biomarker that routinely used as a diagnostic tool to detect the presence of prostate cancer. And in prostate cancer, the disorientations of this cell in prostate gland lead to the leakage of the PSA into the blood vessel. So what happened? This PSA will eventually detected and be increased in the blood of prostate cancer patient. And besides that, PSA also reported to be increased in benign prostatic hyperplasia, BPH. So what is BPH? BPH is an enlargement of the prostate gland. This BPH is not considered as prostate cancer. So this PSA also reported to be not able to predict the aggressiveness of prostate cancer. So based on previous study, PSA screening is found to be lack of sensitivity and specificity. This is why we need a new biomarker that have high sensitivity and also specificity to detect the presence of prostate cancer. So one of the platforms for biomarker discovery is by using exosome. So what is exosome? Exosome actually are a tiny capsule or small extracellular vesicles that can be released by a variety of cells in our body, including cancer cells. So these exosomes originally viewed as a garbage bag that allow the cell to remove the unwanted protein from the cell. And in recent years, many fascinating functions of these exosomes were discovered. One of it is the ability of exosome to act as a delivery vesicle, transferring all their content such as protein and RNA from one cell to another cell, and this eventually allows cell-cell communication. Since tumor exosome can reflect the tumor protein content, this is why exosome become an attractive platform for biomarker discovery. So exosomes are formed through multiple endocytosis that initiated at the plasma membrane. So these exosomes are packed into microvesicle, and this microvesicle will eventually fuse with the plasma membrane and release the exosome into the extracellular space. This is why we can detect these exosomes in our body fluid, for example, urine, blood, and also breast milk. So this exosome eventually will be taken up by the recipient cells. So the content such as RNA, protein will be released into the cells, and these make the recipient cells become cancerous. So it is hypothesized that this exosome induce cancer metastasis in the recipient cells. So this question here, are there any ideal protein biomarkers in the exosome that could be used to predict the aggressiveness of prostate cancer? So based on our previous study, we found out few potential biomarkers that can be used to predict the aggressiveness of prostate cancer. So the purpose of this study is to determine the internalizations of the isolated exosome into the recipient cells, and also we would like to evaluate the involvements of these selected protein biomarkers that we found previously from our previous study, and to see whether these selected protein biomarkers are able to involve in cell invasion and migrations of the recipient cells. So how we are going to achieve this objective? At first, to investigate whether the isolated exosome from metastatic prostate cell line able to be uptake by the recipient cells, these isolated exosome from metastatic prostate cancer cells will be labeled with the fluorescent dye, and then the internalizations of the isolated exosome into the recipient cells will be further viewed under the fluorescent microscope. So once we confirm that this isolated exosome is internalized into the recipient cells, next, the involvement of this isolated exosome in the cell invasion and migrations of the recipient cells will be further investigated. So we hypothesize that the internalizations of selected exosomal protein biomarkers into the recipient cell will facilitate the cell invasion and migrations of the recipient cells. The recipient cells here refer to the non-cancerous, the normal prostate cell line, and also the primary prostate cells. So currently this project is performed by one of my PhD students in UPM MAKNA Cancer Research Laboratory, Institute of Bioscience, Universiti Putra, Malaysia. So besides this project, we are currently focusing on identifying of biomarker for bladder cancer. With that, thank you.